So the ENCODE project set out to understand what each of the letters of DNA actually do. So in the Human Genome Project we got the sequence of letters, but we didn't have functional assignments for each of them.
The ENCODE project really was developed to identify all the genes on the genome and additional elements that would indicate whether it was functional. The HGP, the Human Genome Project, previously has done a very similar function, but I would say it's very much like the Ordnance Survey map, where it gave you the overview of a landscape. But now we have different technologies that we've really investigated this in much more depth, and so it's very much like the Street View, where we're really getting more details to look into the elements of a town, and the houses that are on it are very much like the gene features.
So the GENCODE project was one of the projects within ENCODE, was specifically to focus on the genes. So these are structures within the DNA that specify how to make proteins. But one of the things we discovered was that there's actually a large group of genes which don't make proteins.
So we actually found more genes than we were expecting by detailed analysis. We found over 21,000 genes that actually have the instructions for indicating proteins, and yet we found around 9,000 genes that have no instructions for proteins. And we think with new technologies we probably will find a lot more, as many as 20,000, as many as there are of the other type of genes that encode the instructions for proteins.
So far we know very little about these non-coding genes and what they do. Most of them appear to be involved in some sort of regulation. They include new classes of regulatory structures, such as microRNAs, which regulate some of the processes that define one cell type versus another cell type.
So there is another class of genes that we look at called pseudogenes, which are, a lot of people thought, were junk elements within the genome. But actually, with more investigation, we've found that some of these pseudogenes possibly are resurrected and therefore have a functional role.
So the existence of pseudogenes really reflects the sort of evolution of life, that everything is related back to the first simplest organisms, and some genes evolve as a result of duplication, modification and loss. And so some genes that were duplicates that were used in some organisms are not needed in human and have so fallen out of use.
We found that some of these pseudogenes actually have a function, and there are a recent paper that's highlighted the P10P1 pseudogene, which actually mops up some elements that actually are involved in causing cancer.
So the ENCODE project comes at a really great phase of our development of therapeutic treatments based around stem cells. So we now know how to convert stem cells into certain cell types, potentially manipulate them to cure individual diseases where there's genetic defect. But how do you tell that you've really managed to make, say, a liver cell? Well, the ENCODE project has these reference maps of what a normal liver cell looks like, or what other different cells look like. So they'll become a really important reference to compare your therapeutically designed cells against this reference and understand if they're sufficiently the same that you can use them for therapeutic purposes. The GENCODE project, to construct the set of human genes, the human gene set really underpins the functional behavior of the human genome. So it's completely fundamental to being able to interpret variation in a medical context, which genes are working and which genes have errors in particular individuals. So everything gets mapped back to the genes, which is why it's so important to have a complete set as accurately determined as possible.
I think really with all the different technologies that we integrate, the GENCODE project really is the best gene set that the researchers can use at the moment.